Welcome, and thank you for joining us today. We're excited to highlight a new collaboration between Twist and Element Biosciences. With the launch of Element's Trinity Freestyle workflow, we're unlocking new flexibility in sequencing library preparation, combined with Twist's advanced capture system and probe design. Together, we're trying to push the boundaries of hybrid capture and take on some of the most challenging regions in the human genome. Across the human genome, there are short stretches of repetitive DNA, usually a repeated motif of 2 to 6 base pairs in tandem, so we refer to these repeats as short tandem repeats, or STRs, and over a million STRs have been identified. Due to their repetitive nature, it makes it easier for errors to occur during DNA replication, so the repeat is often expanded or contracted. This feature of STRs means they can have varying lengths in different individuals and can be a source of genetic diversity, and we have leveraged these in forensic sciences to help us in identification. However, if STRs get too long, the region can become unstable and may eventually even impact gene function. When an expanded STR is at risk of being unstable, this is referred to as a premutation, and it's important to monitor as the region is more likely to expand even further in future generations. Once an STR becomes so expanded it impacts gene function, we call this a full mutation or pathogenic expansion. And pathogenic expansions have been associated with over 60 different syndromes, especially neurological disorders. So it's critical that we effectively detect and characterize STRs. Because STRs are highly repetitive, this makes them difficult to resolve with standard molecular tools. The gold standard has long been Southern blot, and this works well for large fragments, but it's manual, low throughput, and increasingly hard to sustain in modern labs. Repeat prime PCR is the next step forward, creating laddered fragments to help you infer allele size. It's certainly more streamlined, but still limited to one or a few loci at a time. That's why the field is moving toward next generation sequencing, or NGS-based approaches, which can assess sequences across the genome. PCR-free whole genome sequencing is showing promising results, especially as longer read lengths continue to improve resolution. And while sequencing costs keep dropping, target enrichment on the exome with short read sequencing still remains a core assay for many labs aiming to consolidate workflows, maintain cost efficiency and feasibility in data handling. Ultimately, our goal is to help you get more out of your exome, enabling detection of repeat expansions within the same assay and reducing the need to perform separate or orthogonal tests to only when you need confirmation or deeper resolution. This is where we see element sequencing chemistry as an ideal fit for pushing the limits of what's possible with short read sequencing. Because there's no on-instrument PCR, we avoid the common challenges of PCR amplification bias. Instead, Element uses rolling circle amplification to generate polonies directly from the original template, improving processivity and sequencing accuracy. The new Trinity workflow integrates hybrid capture with sequencing on board the Avidity. This workflow automates several pre-sequencing steps and combined with Twist fast hybridization system reduces the workflow to a single day. The onboard capture allows us to leverage a fully PCR-free workflow, which gives us the best opportunity to perform enrichment of repeat expansions. Trinity Freestyle gives you flexibility in sequencing library preparation with no special conversion required. Together, Twist has tested our library prep solutions for full compatibility with the Trinity workflow. And importantly, Twist can provide PCR-free library preparation solutions, including enzymatic fragmentation and full-length adapters to make the workflow simple, scalable, and automatable. And with our fast Hybe system, this allows us to bring hybridization time from overnight down to just 1 to 2 hours. The last product I want to highlight is Twist Custom Panel Solutions. At Twist, we don't just manufacture oligos, we focus on high quality performance and consistency at delivery. Our synthesis platform enables uniform oligo production across the full GC spectrum, and we boost probe representation based on GC content to ensure balanced hybridization efficiency, a critical step when targeting low complexity or difficult genomic regions. Every panel is NGS-QC to confirm that probes are present at the expected representation. We can also blend custom content into our fixed designs, like our best-in-class exome. With the help of Twist expert design team, we developed a specific repeat expansion spike-in to pair with the Twist exome. Using Twist EF 2.0 PCR-free library prep solution and fast high buffer system with the Trinity freestyle workflow, we performed exome sequencing on a set of samples previously genotyped and confirmed positive for expanded repeats in clinically relevant genes. We specifically focused on challenging loci such as GC-rich genes FMR1 and C9orf72 and selecting samples that represented a range of repeat sizes. Our goal was to determine whether this workflow could differentiate between normal, premutation or carrier, and pathogenic alleles. The table here highlights a subset of the samples analyzed, giving an overview of the target loci and allele sizes we characterized as part of this evaluation. Even after removing PCR as a factor, we knew this protocol would still require optimization. Low complexity regions tend to form secondary structures, and melting temperatures can vary, especially in GC-rich sequences. We tested several parameters including hybridization, temperature, probe concentration, and even asymmetric read lengths to see if a longer read one could improve alignment. The biggest improvement came from raising the hybridization temperature, which boosted coverage in GC-rich regions. However, maintaining that temperature too long negatively impacted AT-rich regions. Our solution was a step-down hybridization approach, starting hot to denature GC-rich areas, then gradually cooling to preserve AT-rich regions. This extended the hybridization time slightly from 1 to about 2.5 hours, but the trade-off delivered better coverage uniformity and more robust probe-target pairing across the genome. Importantly, we found that our modified protocol and repeat expansion probes did not compromise performance. Results were nearly identical to a standard exome on the Avidity in terms of on-target rate, uniformity, and dropout. Even more exciting, we observed improved variant recall, particularly for indels when compared against genome in a bottle benchmarks. This suggests the workflow may offer better sensitivity in challenging regions and help reduce allele bias while maintaining overall exome quality. The key question, of course, was whether we could actually detect repeat expansions and classify them as pathogenic. And the answer is yes, mostly. Using Expansion Hunter, we successfully classified 7 out of 9 loci, clearly distinguishing normal from expanded alleles. That said, we did observe underestimation of repeat sizes in some cases, a known limitation of short read sequencing and consistent with the original Expansion Hunter publication. Still, the ability to reliably distinguish normal from expanded is a strong step forward, with the goal of only positive or ambiguous samples needing additional confirmatory testing. And here is an example in Repeat Viewer showing detection of both the normal and expanded allele in one of the positive DMPK expanded samples. These two loci, FMR1 and C9orf72, were always going to be challenging. Encouragingly, we did achieve modest read-through coverage and could distinguish the normal allele from a potentially expanded allele. For C9orf72, the pathogenic minimum was within the confidence interval of predicted expansion length, and for FMR1, the premutation range. When comparing the same samples sequenced by PCR-free whole genome, we saw significantly higher repeat coverage and greater confidence in allele sizing. This tells us further optimization and capture will be the focus of our next steps to continue improving accuracy in these difficult regions. In summary, the Trinity Freestyle workflow paired with Twist library prep and FastHype solutions offers a flexible, single-day hybrid capture solution that can be run completely PCR-free, minimizing errors and bias. It delivers performance comparable to traditional exome sequencing while showing improved variant recall, particularly in challenging regions. Using this workflow, we've demonstrated the ability to distinguish pathogenic repeat expansions across multiple loci. Our next focus is to continue optimizing performance in high GC regions to further enhance coverage and accuracy. I'd like to acknowledge the teams who made this work possible. From Element, all of the hard work was really from Kelly and Semyon on study design, protocol, and analysis. And from Twist, the library preparations was done by our Twist Services Lab, led by Kristen and Kate, and input from our research and FAS team, Esteban and Tim. So thank you for all your incredible collaboration and contributions. If you're interested in exploring this workflow for yourself, Twist now offers Trinity for exome kits available for purchase. Our repeat expansion panel is also available upon request. The Trinity workflow may be compatible with other fixed and custom panels, and our team is happy to provide consultation or design support to help you get started. For more information, please feel free to reach out to one of us or your NGS sales representative. Thank you for watching.